In this video lecture, I will be talking on the role of 12 base pair to 23 base pair in TCR gene segments recombination. Random gene segments recombination actually directed by the spacer of recombination signal sequence, which could be, as can be seen here, 23 base pair, which flanks the 3 side of the variable region of the alpha or TCR alpha gene segments, or it could be 12 base pair, as can be seen here, that flanks the 5 side of the joining gene segments of alpha gene, and the gene segment recombination, as I mentioned, that it is directed by the role of 12 base pair to 23 base pair. That means the recombination or the joining, it takes place from 12 base pair to the direction of 23 base pair. So the recombination itself initiated by recombination activity gene 1 or RAG1 and recombination activity gene 2 or RAG2, which will in turn increases the diversity of TCR by random gene signal by random gene segments recombination. So, as can be seen here, these are TCR alpha gene segments flanked by recombination signal sequence number 12 and recombination signal sequence number 23. And this is for the TCR beta gene segments. They are, as can be seen here, the RSS or recombination signal sequence 23 flank the 3 side of the variable beta segment, but however it is it flanks the 23 base pair flanks the joining beta segment, in contrast to alpha, as can be seen here. This is alpha, this is the 5 side of the joining alpha. It is flanked by 12 base pair spacer of recombination signal sequence, but here it is flanked by 23 base pair spacer of recombination signal sequence. For the diversity segment, it is flanked by 12 base pair recombination signal sequence on both sides, as can be seen here. So we expect that the direction here of the joining of the beta or TCR beta gene segments, it will be according to the role of 12 to 23, it will be from this 12 base pair recombination signal sequence that flank the 5 or the 3 side, actually, of the diversity segment. So it will be this direction. That means diversity will bind, or the diversity or D segments will bind to G segments of the TCR beta, and then, as can be seen here, it is flanked on the other side also by the 12 base pair. So following this joining or the binding of D to G, so DJ will bind according to the role of 12 to 23 to the V beta. So the process of recombination of gene segments generated by two enzymes. However, it is directed by recombination signal sequence according to the role of 12 base pair to 23 base pair. But it is generated by two enzymes known as RAG1 and RAG2, which binds recombination signal sequence 23 base pair that flanks 3 sides of variable gene segments and 5 sides of gene segment 12 base pair that encode alpha TCR. And as can be seen here, this is the recombination signal sequence. It flanks the 3 sides of the variable alpha, and the recombination signal sequence, the spacer with a spacer of 12 base pair flanks the J alpha. So the direction of the joining of the segments will be from 12 base pair towards 23 base pair recombination signal sequence. So this will lead to J will bind to the V alpha, as can be seen here. Then the recombination itself is initiated by RAG1 and RAG2, and they come together like this, V1 and J1, V alpha and J alpha, and then the RAG will cut here between this signal sequence and or between the RSS of 23 and below the 12 base pair. It will cut here and it will cut here, and this will end with formation of hairpin. It looks like this. Actually, during this stage, some nucleotides known as palindromic or B nucleotides will be added. Then the hairpin get cleaved, and another enzyme known as terminal deoxynucleotidyl transferase together with some other enzymes, for example Artemis and DNA ligase will start to act, and some N actually, not B, N nucleotides will be added, and this process actually increases the diversity of the TCR and end by formation of functional gene that has the capacity to encode TCR alpha chain of the T cell. For the beta chain, in gene segments encode beta gene of TCR, RAG1 and RAG2, which binds recombination signal sequence 23 base pair that flanks this, as can be seen here. These are gene segments for the beta chain of the TCR, and as can be seen here, the recombination signal sequence 23, it flanks the 3 side of the variable beta. However, in contrast to the TCR alpha or the joining gene segments of alpha, as it can be seen here, it is flanked by 12 base pair spacer recombination signal sequence. Here, the joining segments of beta, they are flanked by 23 base pair spacer of recombination signal sequence, and that is why, because if we look here as a diversity segment, as a diversity or as a D sequence, it is flanked by 12 recombination signal sequence on both sides. So according to the law or the role of 12 base pair to 23 base pair, This diversity will bind to the joining, actually will to the joint segment, and this will lead to DJ, and then DJ will bind to the V, and this, as can be seen here, DJ will bind to V, and ultimately this any of the gene segment bind J gene segment, then DJ binds any of V1 up to maybe there are several B's, and this leads to formation of VDJ and result in a functional gene that has the capacity to encode the TCR beta of the T cell. Thank you very much, Professor Ahmed Bola.